All right, finally, we are on the last lecture. I apologize for having 6 lectures in this chapter, but I guess I could have crammed it into 5, but I decided it would be better to spread it out a little bit more because it is pretty dense, this material. Anyway, in this last lecture, we are going to talk about something called postsynaptic potentials in just a second, and then in a minute after that, we will talk about a little bit more about neurotransmitters. So what are postsynaptic potentials? I feel like the name, it's kind of like a mouthful and it's a little bit daunting, but it's actually really a pretty simple concept. So in the previous chapter, we were talking about synapses, the previous lecture, and we were talking about synapses occurring between 2 cells, but in reality, what you have in the body is a neuron is often receiving messages from maybe dozens of other neurons. And particularly in the central nervous system, I mentioned that one of the job of those tissues is to integrate messages and put them together. So one of the ways that neurotransmitters often act on target cells is by opening chemically gated ion channels. So oftentimes the neurotransmitter will bind to the receptor on the postsynaptic cell, and the postsynaptic cell will respond by opening chemically gated ion channels, and that will create graded potentials. And depending on whether the graded potentials are excitatory or inhibitory, whether they depolarize or hyperpolarize the cell, that can make it more or less likely for the postsynaptic cell to fire an action potential. So these graded potentials that result from neurotransmitter signals are called postsynaptic potentials. So it's exactly what it sounds like, right? So it's a potential, a graded potential, that's happening as a result of neurotransmitter traveling across the synapse. So it's a graded potential that happens in the postsynaptic cell as a result of neurotransmitter activity. Okay, so if the neurotransmitter causes ion channels to be opened in a way that causes depolarization of the postsynaptic cell, that makes it more likely for that cell to trigger an action potential. And we call that an excitatory postsynaptic potential. As you can probably imagine, the opposite of that, if a neurotransmitter causes ion channels to open and that causes a hyperpolarization of the postsynaptic cell, we call that an inhibitory postsynaptic potential. That's going to make it more difficult for that cell to generate an action potential by making the membrane potential more negative, so you have to work harder to get to that threshold voltage. Okay, so here is just a visual kind of going over the same thing that we just talked about. So here you have an EPSP. This is going to be an excitatory postsynaptic potential because as a result of neurotransmitter signaling, you've opened an ion channel that caused depolarization and brought it closer to the threshold. It's still not there, right? You need something else in order to trigger an action potential, but you brought it closer. It's excitatory. In the opposite case, an inhibitory postsynaptic potential, you made the inside of the cell more negative, so now it's going to be harder to fire an action potential. Okay. Just like when we were talking about graded potentials and how they can be summed, so can postsynaptic potentials be summed, added together. So there's 2 different ways that these postsynaptic potentials can be summed. There's what's called spatial summation and what's called temporal summation. So spatial summation is summation over space, over area. So we can see here that we have this one postsynaptic neuron, which is labeled number 3. It receives messages from 2 other neurons that are labeled number 1 and number 2. So we can see here, oops, I'm sorry, I did not mean to skip ahead there. All right, so neuron number 1 releases neurotransmitter and causes an EPSP in cell 3, and that's shown right here. Cell 2 does the same thing, fires an action potential, releases neurotransmitter, and that causes an EPSP, and that's here. So if they are doing these 2 things separately, it goes back to baseline after the first one, so there's no additive effect. But if they both fired at close to the same time from different areas, they can get added together. And now the combined effect of both of these EPSPs gets you to the threshold and triggers an action potential. So if just one fired, you wouldn't get there. If just 2 fired, you wouldn't get there. But if you're summing across both of them because they're happening at about the same time, then the additive effect can cause number 3 to fire an action potential. Okay, this can also happen if you have a single presynaptic neuron, and this is called temporal summation, summation over time. So you can see here that it's kind of the same scenario happening in these first two instances where this presynaptic neuron is releasing neurotransmitter, it's causing an EPSP in the postsynaptic neuron, but you're not getting up to the threshold for triggering an action potential. Happens again, not sufficient. But what happens is if the presynaptic neuron starts releasing neurotransmitter rapidly in response to many frequent action potentials, what can happen is that the EPSPs can get summed up, because after the first one happens and ions flow in, it hasn't gone back to baseline yet, and more ions flow in from the next action potential, and then more ions flow in, and then more flow in, right? And eventually you get to the threshold for triggering an action potential, and that's shown right here. So that's what summation is. So spatial summation is across two different cells, and temporal summation is summation of signals coming from a single cell, but very close together in time. Okay. So then I wanted to spend the rest of our time talking a little bit about neurotransmitters. So we don't have a lot of time to go over this. I'm going to talk about them very briefly, but there are a few different classes, and I just wanted to mention each of them. There's many dozens of different neurotransmitters that exist in living things. But the 7 basic classes that exist in animals are going to be, first there's one called acetylcholine. That's a single neurotransmitter that is actually its own class all by itself. We will talk about that in a couple weeks when we talk about control of muscles, because this neurotransmitter is responsible for controlling the activity of muscle contraction. So that's obviously important. There are then the biogenic amines. So those are going to include things that you may have heard of before. So the neurotransmitters serotonin, dopamine, norepinephrine, and epinephrine, I know you've heard of before. They're clumped together because they have similar structure. Interestingly, because they have similar structure, most of them have some affinity for the other's receptors. So they have some ability to have overlap between all of them. These neurotransmitters happen to play an important role in mood, and a lot of them are going to be targeted by various psychiatric medications. And one of the reasons why psychiatric medications are notorious for having a lot of side effects and for having a lot of unintended consequences, one of the reasons for this is because there's not a lot of specificity of these neurotransmitters, so they tend to have overlapping function, and they affect things other than just treating the underlying psychiatric issue. All right, there are also amino acids that act as neurotransmitters. These are particularly important within the central nervous system. And they tend to be classified based on whether they cause EPSPs or IPSPs. So we call them excitatory or inhibitory neurotransmitters. The most important and well-known inhibitory neurotransmitter is something called GABA. And the most important excitatory neurotransmitter is called glutamate. Neuropeptides. So these are going to be peptides that are secreted from neurons. They're going to be important in a lot of pain sensing pathways. There are also purines. So remember when we were talking about purines and pyrimidines back in Bio 151, there are actually some nucleic acid derived molecules that act as neurotransmitters. There are gases that act as neurotransmitters. One of the most important ones is NO, which stands for nitric oxide. So nitric oxide is important for smooth muscle relaxation. You might actually remember when we were talking about the cardiovascular system, I talked about the fact that a drug called nitroglycerin is often given to treat angina or chest pain. So the way that this works is that nitroglycerin is very quickly metabolized in the body into nitric oxide, and it acts on the smooth muscle in blood vessel walls to open up blood vessels leading to the heart, so it improves blood flow to the heart, which treats the chest pain associated with angina. There are also lipids, so that's going to include what are called endocannabinoids. You may have heard of endocannabinoids before. The term cannabinoid sort of might sound like something familiar. Cannabis, if you've heard of it. So cannabis is the name of a plant that is used recreationally to produce feelings of I guess a high, you could say. And the way that endocannabinoids work is that they bind to, these are going to be neurotransmitters that bind to receptors, And these same receptors also get excited by exogenous cannabinoids, cannabinoids that come from the marijuana plant. So that's why we have that ability to respond to marijuana, is because it actually mimics the effects of these endocannabinoids that we have naturally in our body, but it activates them much more robustly. Okay, so on that note, I did want to just end by introducing you guys to a website that I think is very fun and interesting. It's called Mouse Party. So we're going to link there now. So this is a website that's run by the University of Utah. And if you try to access this from a phone, I'm just going to caution you that it uses Flash. So depending on what kind of phone you have, it might not work. But it's kind of cool. I'll just introduce you to it really briefly, and then if you want to explore it more on your own. So I'll just let you watch the animation. So the premise here is that, it's a mouse party, and each mouse is using some sort of different recreational substance. And what you're going to do is you can take each mouse. So why don't we take the ecstasy mouse, and you can drag it into the chair. And it tells you a little bit about how that drug affects the brain.
Serotonin transporters are responsible for removing serotonin molecules from the synaptic cleft after they have done their job. Ecstasy mimics serotonin and is taken up by serotonin transporters. In fact, ecstasy is more readily taken up than serotonin itself. This interaction with ecstasy alters the transporter. The transporter becomes temporarily confused and starts to do its job in reverse. The excess serotonin becomes trapped in the synaptic cleft. As a result, it binds again and again to the receptors, overstimulating the cell. Ecstasy affects serotonin pathways responsible for mood, sleep, perception, and appetite. Ecstasy also indirectly interacts with the reward pathway. The excess serotonin stimulates a milder release of dopamine along the reward pathway, giving ecstasy slightly addictive properties.
All right. Anyway, I think you get the picture. You can explore that on your own time if you'd like. But I just always like to put that out there because I do think it's fun and it's a reliable source of information. All right.